Hello, I'm Andrew Phillips. I'm going to be talking to you about measures of disease, and the first part of this lecture is measures of disease risk or burden. The areas to be covered in part one of this are to define the prevalence, the odds, the risk, the rate, and Kaplan-Meier estimates. So why are these measures of disease important? These measures form the building blocks of epidemiological research, helping us to understand patterns of disease occurrence, risk factors and potential causes for disease, prognosis of disease, and how to plan for health services. Now, these measures are frequently used. I looked in the Web of Science to see the number of times that these terms had been referred to in scientific articles in 2019, and you can see that there's many thousands of times. So it's very important that you understand the definitions of these terms. All of these measures have been employed in various aspects of research on COVID-19, for example. Now, you've heard about the different types of outcomes that are going to come up in this course, and this lecture concerns the second and third of those, binary outcomes and binary time to event outcomes. So for our measures of disease or risk or burden, we can consider what are the types of outcome measure. So for the prevalence, that's for a binary outcome measure, the same with the odds and the risk, all for binary outcome measures. Whilst a rate or Kaplan-Meier estimates that we're going to be talking about, they would be using time to event outcomes. So the first definition that I want to show you is that for the prevalence. So the prevalence is the proportion of persons in a population who have a particular disease or attribute at a specified point in time. So the denominator for this is the number of people in the population at that point in time, and the numerator is the number of people in the population with the disease at that one point in time. And it's usually expressed as a percentage, so multiplied by 100%. Prevalence is a measure that's particularly relevant to describing burden of a chronic condition. Cross-sectional studies, which you've heard about, are one type of design that allows calculation of a prevalence. So let's consider an example. Prevalence of cytomegalovirus infection in young adults. So let's imagine we have 50 people aged 18 living in one country, and they were tested for presence of CMV antibodies, and 27 of them tested positive. So our estimate of the prevalence from that sample would be 27 divided by 50 times 100% gives 54%. So let's think about the interpretation of that. Does this mean that 54% of all 18-year-olds in the world are infected with CMV? Well, no, clearly it doesn't. So 54% is an estimate based on our sample. And we're uncertain about whether our estimate of 54% prevalence of CMV for all 18-year-olds in the world is correct, for two main reasons. The first one is that the sample is not a random, unbiased sample of the population to which we wish to generalize our results. So the characteristics of the population from which we sampled may not be representative of all 18-year-olds. For example, we are only sampling from one country. The second major reason is that the role of chance due to a limited sample size. So even if we had a truly random sample of 18-year-olds from across the world, we only have 50 people in our sample. If we took two random samples each of 50 people, we would generally not get the same estimate. Now, for the first of these sources of uncertainty, we generally don't actually quantify this. When presenting the estimates, we state that it's uncertain if the estimate applies outside the population sampled. The second, though, the role of chance, we quantify this uncertainty by giving a 95% confidence interval around our estimate, which you've heard about already on this course. So let me remind you of the definition of a 95% confidence interval. So we conceive of what value we would have got had our sample been of infinite size, which I'll just call the infinite sample value. So this infinite sample value is usually called the true value, or the value in the population from which we've sampled. Now, 95% confidence interval around an estimate is the interval within which we are 95% confident that this true value lies. So note that although it is standard terminology, such terms as the true value can be misleading because they might imply that our sample is a representative, unbiased sample of the population, when that's not necessarily the case. So going back to our example here, where we have the prevalence of CMV infection in young adults, the 95% confidence interval based on 27 out of 50 is 39.9% to 68.1%. So this means we're 95% confident that the true prevalence of CMV lies between 39.9% and 68.1%. And remember, this is taking into account only the second of those sources of uncertainty that I mentioned. Now, if we increase the sample size, this will narrow the width of the confidence interval. So if our sample was 100 times larger, but the prevalence was the same, so 2,700 out of 5,000, the 95% confidence interval comes to 52.5% to 55.5%. It gets narrower as the sample size gets larger. So next, the odds. The odds of having a particular disease or attribute is given by the number of people with the disease on the numerator and the number of people without the disease as the denominator. Now, odds are mainly used in the context of case control studies. So to take an example, going back to the CMV infection in young adults that we were talking about earlier, the prevalence of CMV, if you recall, was 0.54. The odds of CMV is 27 with CMV divided by 23 without CMV. So that's an odds of 1.17. So now we're going to be talking about measures of disease relating to incidence of disease events in people followed over time. I'll be talking about the risk, the rate, and Kaplan-Meier estimates. These measures are typically calculated from cohort studies or other studies involving follow-up of people, such as clinical trials. So I'm going to be asking you just a couple of questions, which will come back to the answers in part 2, where we illustrate some of the measures of association that we come onto in the next part. So the first question is, if a person with HIV is successfully treated, does this affect how likely they are to transmit HIV if they are having condomless sex with an HIV negative person? Have a think about which of those three answers you think is correct, and I'll show you the answer in the next part. The second question is, if an HIV negative person takes antiretroviral drugs, can this make them less likely to acquire HIV when they're having condomless sex with an HIV positive partner? And again, there's three possible answers, and just give some thought as to what you think is the answer. And I'll show you some examples of a couple of studies that have addressed these two questions, which illustrate the kinds of measures I'm talking about. Now, in order to be able to illustrate data of this type, this figure shows data obtained on people followed over time. So we have 22 people represented by these 22 lines followed for 2 years to ascertain if and when an event, which may be a disease or death, occurs. So the lines are numbered 1 to 22. The x-axis shows up to 2 years of follow-up, and the line shows a person being followed over time. And if they develop an event, then the line stops at that point, and the event is indicated by a diamond. So here we define the first measure, the risk. The risk will be generally familiar to you. It's the number of people experiencing the event in a defined period divided by the number of people in the study population at the start of the period. Now, risk is a number between 0 and 1 and can be expressed as a proportion or as a percentage, and it must relate to a specific time period. So in our example data that I showed you on the previous slide, we had 22 people, of whom 5 developed an event. So the risk of an event by 2 years is 0.23. So here's an example of an article which has used the risk measure. This was an article about pre-exposure prophylaxis for HIV prevention in men who have sex with men, and it relates to the question I asked earlier. So in the placebo arm for this study, there were 1,248 HIV negative men followed. 64 of them became infected with HIV over up to 132 weeks of follow-up. So the risk was 64 divided by 1248, 5.1%. And they calculated a 95% confidence interval for that in the paper. So I'm going to introduce the rate to you, but in order to be able to do that, I need to first introduce the person years at risk. Person years at risk is the sum over all people of the number of years in which individuals at risk of the event have been followed. So person years are calculated for time to event data. So how do we calculate the person years at risk for a sample of people? Going back to our 22 people here, you can see down the right hand side I've written the number of person years at risk that each contributes. So a person that's followed for the whole 2 years and doesn't develop the event contributes 2 person years at risk. They were at risk of developing the event over those 2 years but didn't in fact get it. Person number 4 here though, who did get the event, got the event after 1.3 years. They spent 1.3 years at risk of getting the event, and then they did get the event. After that, we don't consider their time as person time because we're interested in person time at risk of having the event for the first time. So the people that got the event, their person years at risk are only counted up to the time of the event. Now we then sum the person years at risk over the whole sample. So this is a concept that you're probably not really used to, but we sample the person years at risk over the whole sample, and in this case it comes to 39.2 years. So in total, in this sample of people followed over time, we have 39.2 years of experience in which people lived at risk of the event that we're interested in. So then how do we define a rate? Now a rate is sometimes called an incidence rate or just an incidence. So a rate is a measure of the rate with which an event, such as a new case of disease or death, occurs in a population over a period of time. So the definition of a rate is the number of people experiencing the event in a defined period divided by this person years at risk that we've just calculated. So in our example data, the rate is 5. It's the same as the risk. There's 5 people who developed an event during the follow-up. But the denominator now is the person years at risk for the group. And that in total was 39.2 years. So dividing one by the other comes to 0.13. And this has units of per person year. So it's 0.13 per person year of follow-up is the rate experienced by this group. So the unit for the rate may be expressed per 100 person years or per 1,000 person years or for any time period. So for example, the rate of HIV infection in men who have sex with men in the UK in 2015 was approximately 0.6 per 100 person years. A rate will always be a positive number. If the event of interest is death, then this is called a death rate or a mortality rate. The rate takes into account when events occur during the follow-up period. This kind of data, where we have the time period before an event occurs, are often referred to as time to event data, time to failure data, or survival data. And we call them survival data even if the event of interest is not actually death. So let's now consider a comparison of rates and risks. On the left-hand side here in this example in the blue, we have 8 people followed over time, of whom 4 developed an event. So the risk in 10 years is 50%, 0.5. And over the right-hand side, similarly, in this example, 4 people developed an event over a 10-year follow-up, 50% risk. But if we calculate the rate, the person years over the left-hand side is 76. The person years over the right-hand side is only 44. So the rate over the left-hand side is 53 per 1,000 person years, whilst over the right-hand side it's 91 per 1,000 person years. So in a situation where risks are the same, the rate can be very different. So censoring. By using person years at risk as a denominator, as we do in a rate, we can accommodate the fact that not all people are followed for the same period of time. If a person is no longer followed for any reason, we refer to the time to the event as being censored at the end of the individual's follow-up time. And we use the term censored because we can't see it. So here the data in our example have been adapted so that we have the same people developing an event at the same time, but we have other people for whom the follow-up is censored at a certain point in time. They are no longer being followed. And so if they were to develop the event, we wouldn't ascertain that event. And so we can't count those as person years at risk. So with the green dots here, that is when the person time for each person is ending due to being censored. So you can recalculate the person years at risk by adding the numbers along the right hand side, and it now comes to 34.7. So the event rate now has changed to 5 divided by 34.7, which is 0.14. So now you know what a rate is. I'll tell you about this paper which looked at prevention of HIV infection with early antiretroviral treatment. 882 HIV seronegative people who had an HIV positive partner were followed for 1,567 person years. 27 of them became infected with HIV. So the rate of infection was 27 events, HIV infection is the event, divided by 1,567 person years at which people were followed at risk of HIV. And that comes to 1.7 per 100 person years of follow-up. And they presented a 95% confidence interval. In the second part, I'll show you the comparison group. Now, a way that we commonly see that time to event data that we've been talking about are summarized is something called a Kaplan-Meier plot. And this is a Kaplan-Meier plot of the probability of having a disease by a time of follow-up. So along the x-axis, you have the years of follow-up, and along the y-axis, you have the probability of having had the event by that time. So Kaplan-Meier plots are drawn as a series of steps in which only changes at the time that someone develops an event. That's where you see a step, that at least one person at that time point has developed an event. So this is a way of summarizing our time to event data on the 22 people followed in our sample. And you'll see this kind of plot commonly in journals when you read articles, in particular randomized trials comparing 2 different groups. You'll see these Kaplan-Meier plots for the different groups. I'd also like to point out the interrelationship between prevalence and risk that we've talked about. So high prevalence of a disease or condition may reflect the fact that there's a high incidence of that disease, a high risk of it occurring. It can also reflect the fact that there's a prolonged survival without a cure, so a person who gets it has it for a long time. A low prevalence can reflect the fact that there's just a low risk of developing the disease in the first place, or it can mean that there's a rapid fatal disease progression or a rapid cure. So it may be relatively common for someone to get it, but they won't have it for very long, and that can be consistent with a low prevalence. So it's important to think about the interrelationship between prevalence and risk, and they're not necessarily correlated. So that's the end of part 1 of this lecture. Thank you for listening. In the second part, we're going to be talking about measures of association between exposure and disease and revisiting these measures of disease that I've talked about.